I am Dr Mootaz Salman. I'm based at the University of Oxford, and I'm part of the BHF-UK DRI Centre for Vascular Dementia Research. My research program evolves around understanding the role of a blood-brain barrier in neurodegeneration. This is an intracellular interface that aims to protect the brain and provide the optimum environment for the neuronal function. However, it's a double-edged sword because it stops about 98% of all the new drugs from getting into the brain. So my group and I will be focusing on understanding the mechanisms involved in improving the transport of the new targets and the drugs, but as well as understanding the role of this particular intracellular interface in neurodegeneration through understanding the mutations and also risk factors, particularly of importance for vascular dementia research. Well, stem cells are an invaluable resource for us, in particular for studying neurodegeneration. If you want to study neurodegeneration, you are working on the brain, one of the hardest organs to get access to. You cannot just stop someone down the street and say, hello, can I use part of your brain? So the way that we're currently relying on is we get in cells, either from autopsies, which means patients undergoing other surgeries, or from postmortem tissues, or from patients who've been passed away. Either way, these cells are not in an optimal condition to interrogate the system or to do tests. With these particular stem cells, now we can start from the exact same resource to differentiate all the components that I need to build a humanized model of a blood-brain barrier that is relevant for vascular dementia. Drug development is such a long and expensive process. From the moment that it's an idea in someone else's head until you see it on the shelf by Boots, it's about $2 to $3 billion. Now, the success rate of that is anything between 5 to 10%. So the reason for that is because the majority of the drug, in fact, 80% of them, that they pass the preclinical animal models, they will fail in the phase 1 human clinical trials. And the reason is simple, because we're different. That's why the use of the humanized research should hopefully address that gap. More importantly as well, refine and use the animal in research whenever they are not needed.